All right. We have a few kind of straggler I can statements, learning targets that I'm going to put together. And the first is I can relate the structure of the mitochondria to the function of completing the citric acid cycle and oxidative phosphorylation. And so if you look at the structure of the mitochondria, it has an inner membrane and it has an outer membrane. It has that in-between space. Well, the citric acid cycle occurs in here. It's going to be producing a lot of those NADHs and FADH2s, the ones that are produced in pyruvate oxidation, which happens in this inner membrane space, and glycolysis, which happens out here, will get shuttled into the matrix. But if you picture, let's make ATP synthase. Okay, if you picture oxidative phosphorylation, that looks like a bladder or something. It's not. It's ATP synthase. You're pumping hydrogens from the matrix to that inner membrane space. So you're pumping hydrogens from the matrix into that inner membrane space. So it makes sense. And if you also remember, the NADHs were on the matrix side. So it would be good for us, efficient is a good way to put it, if a lot of those NADHs are already here, and that's what's happening. They're being produced in the citric acid cycle in this matrix. So they are already in the matrix space or area to do what they need to do. Also, as I mentioned earlier, you have to have a membrane in order to create that gradient. So we have lots of hydrogens. We have high hydrogen ion concentration here and low here. The membrane is what allows us to have high and low. If there was no membrane, they would just all mix up and it would just be even. Diffusion would happen. It would just be even. And so this selectively permeable membrane allows us to create that high concentration. And so that's how the structure of the mitochondria really gives, makes it easy, I guess, makes it more efficient for the citric acid cycle to then lead right into oxidative phosphorylation. The next one says, I can identify where each of the 6 carbons are broken off of glucose. So let's look through the different phases or different steps. This is glycolysis. We didn't lose any carbons there, so none were broken off there. We lost 1 here, but that's actually 2. So remember, glucose is a 6-carbon molecule. So we lost 2 carbons here. And then we lose, if you look down here in the products, per glucose, the other 4 here. So by the time we get through pyruvate oxidation and the citric acid cycle, we have lost 2 carbons here and 4 here for a total of 6. So at this point, by the time we get to the end of the citric acid cycle, we have lost all 6 carbons. The next one says, I can explain how the breaking of a bond leads to the reduction of NADH. And so we can look at this really in any of these, but we'll go back to glycolysis. When we broke that bond, it released electrons. Those electrons carry energy, and they go to NAD plus to make NADH. And so by breaking that bond, you allow those electrons to be reduced, and they're given to NADH. And the last one says, I can identify the locations of cellular respiration in which ATP is produced. So we're producing 2 here in glycolysis. Oops. None in pyruvate oxidation. Okay, so none down here in the products. 2 in citric acid cycle. Those were produced over here in that last step. And then the vast majority of them, those 34, were produced in oxidative phosphorylation by ATP synthase. So glycolysis, citric acid cycle, and oxidative phosphorylation are the locations in which ATP are produced. So that's those last 4 little I can statements or learning targets. If you still have questions about those 4 or just want me to go over anything again, just let me know. And I hope that helps.